Hello and welcome to another episode of Old World Agriculture. In this episode we'll talk about the domestication of canary seed. Canary seed belongs to the species Phalaris canariensis. Phalaris canariensis is the only member of a 22 species genus in which the ripe seeds are retained within the spike of the seed pod. There's been several hypotheses on the origin of this species. Canary grass, which is a plant we get canary seed from, is one of 3 species that have 6 chromosomes in the genetic set. Amongst them, only 2 are self-pollinating and annual. This is Phalaris canariensis and Phalaris brachyacus. The third one is a perennial that's cross-pollinating, is called Phalaris truncata. Proper hybridization with that species and Phalaris canariensis was attempted, but failed, indicating that it's unlikely that Phalaris canariensis is a descendant of Phalaris truncata. The differences between both species can be seen on the above image. To determine if hybridization between these 2 species is possible, and by proxy showcase a possible link between the 2 species, indicating that one was domesticated from the other, hybridization experiments were done. First, the pollen was destroyed on the canary grass plants by use of hot water. Then, regular hybridization between 2 species was made, and the 3 generations that followed were analyzed. Only a small number of the F1 hybrids produced seeds. The reason why will be discussed in the second part of the video. Amongst the many crosses that were made, only 25 plants were made, 22 that are likely self-pollinated, and 3 that received the dominant gene for seed shedding from Phalaris brachyacus. These 3 were made to self-pollinate in order to create the F2 generation. In the F2 generation, several of the key features began segregating in a 3-to-1 ratio. 67 plants had short florets associated with the wild species, while 15 had long florets associated with the domesticated species. Those with the long florets retained their seed, while those with the short kept them. A great deal of variation between plants with varying degrees of tillering, with higher tillering associated with the wild species and lower tillering associated with the domesticated species, appeared, with most plants showing varying degrees of wild and domesticated traits. From a seed setting time perspective, flower setting time perspective, and so on and so forth. 12 long floret plants from the F2 generation were collected and were allowed to set seed. All 250 F3 plants are derived from these 12 plants that self-pollinated. In the 3rd generation, the difference between long floret and short floret plants sorted itself out into a 1 to 2 to 1 ratio for segregation. This showcases that the major difference between the parental species is controlled by 2 genes at one locus. Now, what can we conclude from this information? What the researcher I'm referencing concluded was that both Phalaris brachyacus and Phalaris canariensis are both the same species, with the cultivated form derived from mutants with a single dominant and a single recessive mutation, with all the variations that show up being derived from cultivation of this mutated form. Although canary seed is still a minor crop within agriculture, work on improvements still being done, using mutants that reduce the hair number on the seeds, as well as other selective breeding projects, CDC Maria being one such example. The crosses were very difficult to make, likely because the method of emasculation damaged the ovules, preventing seed set in certain instances. From the perspective of improving plants, male sterile hybrids between Phalaris canariensis and Phalaris truncata were made, and a backcross back into Phalaris canariensis was also made, using canary seed pollen to pollinate that hybrid. Dr. Goose hoped to use it to make improved perennial grasses for grain purposes, or new forage species that are suitable for alkaline clay soils in parts of the Mediterranean region. These studies also showcased that Phalaris truncata could not be the wild ancestor. If it was, hybridization would be easier. Most of the differences found in the wild form of the species appear to be generated by fixing various alleles into the pedigree of a landrace line, with most of these traits being controlled by multiple genes. This was confirmed by information found in the F2 generation, which showed varying degrees of wild and domesticated traits. Based on current information, there is a limited genetic variability within canary grass. Indeed, many cultivars were studied, and they were all found to be very similar, phenotypically speaking, both in terms of seed size, seed shape, oil, protein, and crude fiber concentrations, and many other traits. What this means in practice is that, in order to increase the genetic variability, crossbreeding with the wild ancestor of canary seed, Phalaris brachycachus, would be necessary in order to broaden the gene pool and improve the traits within the gene pool of canary seed grass. Well, that about covers everything. Thank you for watching. Thank you for watching this video. If you're watching on YouTube, please subscribe to me on BitChute for a greater variety of content, 4 videos a week. And thank you to all my subscribers on both platforms. I appreciate it.